I really should get you to do my PR for me, because that was extremely generous. When she says golden age of science communicators, what she means is not quite as famous as Brian Cox. So yeah, I am a science communicator, which is a weird sort of term, but I'm a journalist and broadcaster. I was a geneticist before that. I was a researcher and worked on genes involved in eye disease and eye development. And so some of the things I'm talking about tonight, which are included in the book, but are also the sort of backbone of the project that I'm working with David Blandy on right here at Lighthouse, are just the themes that emerge when we start thinking about what we know about DNA and what we can do with DNA. And so what I'm going to talk about tonight is part of the history, so how we got to the point where we know this much about evolution, DNA, genes, and inheritance, but also what we can do with that information and what we can do with that technology. And I think these things are, I think in science communication it's important to tell stories. And so when we think about DNA, when we think about evolution, basically what we're talking about is the transfer of information across the generations. Now that's a very clinical way of talking about inheritance. For the longest time, until the 20th century, until the second half of the 20th century, what we knew about inheritance was based on either farming or on families, passing traits through families. And the best studied families were royal families. So I like to start, it's almost an aside, but it's such a good story, I like to tell it as often as possible. I want to start with this guy. This is Charles II of Spain, Carlos. He was the last ruling member of the Habsburg dynasty. He died in the beginning of the 18th century. The Habsburg dynasty had ruled the largest chunk of Europe for 200 years until his death. He died childless. During that time, they had supplied every Holy Roman Emperor for 200 years. Now, Charles was a tragic figure. He was beset by all sorts of physical and mental difficulties. He didn't learn to walk until he was 6, and he didn't learn to talk until he was 8. He had an abnormally large tongue, which was one of the reasons for that. He was almost certainly sterile. He died childless. It was the end of the dynasty, despite many attempts by the family to mate him with various individuals. For that reason, his nickname was, forgive my Spanish accent, I think it's El Jefishado. Anyone want to correct that? No. Okay, so he was known as Carlos the Hexed. Right, now, here are some of the members of his family. That's Carlos on the far left there. Now, they weren't an attractive bunch, but they certainly were a powerful family. They all have this characteristic jaw and lip, which is known as the Habsburg jaw or the Habsburg lip. Medically, it's more accurately known as a prognathous jaw. But although hideously unattractive, this is actually considered to be a mark of this royal family, so something that was desirable. And this is the pattern of inheritance that is why we know about how what we now know are genes and DNA are passed from generation to generation. Now, the reason that Carlos was so hexed was because this family indulged in a really rather impressive level of inbreeding over several generations. Now, I don't know whether you're familiar with what a family tree normally looks like, but this one is Charles's, right? Now, normally, if you go back 7 generations, if you go back 7 generations on a normal family tree, it should only branch outwards. Actually, that's not quite true. It's not quite true, that, because humans are a very inbred species, and so when you go that far back, it turns out that you're probably mating with someone who's already in your family. Statistically, you're sitting within 6 seats of your, I think it's your 7th cousin right now. Anyway, there should be, if you go back 7 generations, 254 members on this family tree. There are 29 people on this tree here. You can see, have I got a point? Is there a pointer? Or I can just point here. So this is Charles here, right? And this is his mother, and this is his father. This is his mother, that's her mother. So that means that Charles's grandmother was also no, wait a minute, let me get this right. His aunt. So his grandmother was also his aunt. And if you go back another generation, so this is his mother, this is his grandmother, this is his great-grandmother, this is his father, This is her mother, so his grandmother was also his great-grandmother. Okay. Family trees shouldn't have loops in them. They just never, that's a bad thing to happen. There's one here, there's one here, there's one here. You can't really start describing generations in this family tree because they're so intergenerational. And if that weren't the worst of it, if you look at the top, you probably can't quite make it out. Joanna of Castile is the woman 6 or 7 generations back, 6 and 7 generations back, more accurately. And at that level, if it was fully outbred, you'd have 32 positions for that woman. Wow. And in the one back from that, 64. She occupied 8 on the 6th generation and 12 positions on the 7th generation. And if that weren't bad enough, her nickname was La Loca, which in Spanish means the insane. So, not a good genetic stock to start from. Inbreeding, very bad for families, extremely interesting for geneticists. Right. So I'm going to jump around in time in order to tell these stories. I'm going to move to a completely different family tree now. Probably the most important family tree ever drawn. This is a sketch that Charles Darwin put in his notebook in 1837, 20 years before he published The Origin of Species. But it's the first time that he began thinking about evolution as being a tree-like structure, how one species would diverge into 2, and that being the basis for the breathtaking diversity of life on Earth. It's nice, I like the way he just writes at the top, I think. He doesn't really finish that thought, but I think, yep, you thought right. This is what the Tree of Life looks like today, or in 2003 at least, the last time it was done in such a comprehensive way. It's called the Hillis plot. And this is representative. You can't quite make them out because of the details here, but each one of these little lines here is an individual species. I think there's around 6,000 on here. Science has described around 2 million species, so this is just a representation of what's alive today. We also know that 97 to 98% of species that have ever existed are already extinct. Right? So this is just 3%, this is a proportion, a fraction of the 3% of extant species. So the tree is big. It's really big. And just in case, so see where it says animals up there and there's a little note below it? That's you. Now, Darwin's tree was being formed in the 1830s, and then he published The Origin of Species in 1859. At almost exactly the same time, this chap, Gregor Mendel, that everyone learnt about in school, was working out the method, the rules by which information is transferred from generation to generation. He did an experiment. He did many experiments on pea plants predominantly. He is invariably described as a monk, and this is something which I've got a slight revisionist goal here. He definitely was a monk. That is factually correct. He was one of the most important scientists of all time. So I always think it's a bit weird to describe him as a monk. Because there's no real record of how good he was at monking. Anyway, what he interbred and inbred thousands of pea plants, looking at individual characteristics in these pea plants, such as flower color and wrinkliness of the skins of the peas, and he bred them together and he came up with 2 rules of inheritance, the patterns by which we transfer information from generation to generation. And those rules basically say that we get one version of each characteristic from each parent, and that when they meet, they don't blend, you just get proportions of those individual characteristics. So if you take a purple flower and you cross it with a white flower, instead of getting whitey purple flowers, sort of pinky flowers, you get a proportion of purple flowers and a proportion of white flowers. Okay? So you get each trait individually inherited. And what he was prefiguring, although he didn't come up with a name, was the concept of genes. Again, with our 21st century understanding of genes, genetics, and evolution, we now know that genes are made of DNA. And Darwin had no idea about DNA. He also had no idea about genes during the whole of his lifetime. By another coincidence, well, Mendel was doing this at the same time that Darwin was working out evolution, by another coincidence, DNA was being isolated for the first time at exactly the same time in Germany, also unknown to Mendel or Darwin, by this guy, Friedrich Miescher. He was a doctor working in Tübingen in the 1860s. And this is a bit gross, but the Franco-Prussian War was going on at this time, which meant that he had an unending supply of pus-soaked bandages. And pus contains leukocytes, a type of white blood cell. And leukocytes have particularly big nucleuses, and nucleuses are what contain DNA. So he was getting all these leukocytes out of pus-soaked bandages, and from that extract, isolated a chemical which contained phosphates. Now, at this stage, we knew a lot about proteins and amino acids. And they don't contain phosphate. So he discovered this new chemical which had a lot of phosphate in it, and he called it nuclein because it came from the nucleus of these pussy cells. But what it was, in fact, was DNA. His work was not really followed up for various reasons, but in the 20th century, it became the iconic and the most important and most famous story in the history of biology, possibly the history of science. So I'm going to quickly whip through that. We don't really need to go into details of that story, but it begins, or it involves, Rosalind Franklin, who, working with Raymond Gosling and Maurice Wilkins, produced the data that Crick and Watson would use to determine that the structure of DNA was the iconic double helix, the most famous, possibly the most famous structure in science. So beautiful and so iconic and so elegantly constructed, it never looks like that, right? It just never looks like that at all. DNA, it's a beautiful thing to have, and it's a beautiful thing to understand that this structure is fundamental to the way it behaves, which means it's fundamental to life, which means it could not be more important in terms of being a molecule. But it just never looks like that. DNA is an incredibly dynamic and active molecule. It is in all of our cells, and in fact all cells that have ever existed. It's always twisting and turning and unwinding and twisting back on itself. It's nice to have this, but when you extract it in a lab, it kind of looks a little bit like snot. Now, why is DNA important? Why is this structure important? It is incidentally the 60th anniversary of the publication of that paper in April. Now, it's important. The structure is important because it does two things, two very, very clever things. The first is that, because it's a double helix, right? If you think about it as a twisted ladder, where the rungs are made up by pairs of chemicals, one attached to each one of the struts. There are 4 of these chemicals. They're called nuclear bases or letters. And they are A, T, C, and G. They pair up on each one of the vertical struts in a very specific way. A only ever pairs with T, and C only ever pairs with G. Now, what that means, because this is a double helix, is if you split it in 2, like this, you have all the information on an individual strand to replace the missing strand. Okay, so where there's an A, you can put a T. Where there's a C, you can put a G. Right? And that means that you can replicate this. It means that you can take one molecule, you can rip it in 2, and then you have 2 molecules. And if you think about the tree of life again, that is how information is passed down. That is how genes work, it's how inheritance works, and it's how all of evolution for the last 4 billion years works. I said about how DNA is never as pretty as it looks in that double helix. This is an animation of that process in action. So in here you have the double helix coming into this structure here, which is called a replication fork, and it splits it in 2. And out of one side you get one single strand, out of this side you get the other single strand. And as it whips around, it goes to another protein complex here, which replaces the missing strand. So, oops, sorry. Which means that this bit coming out is the second double helix, and this bit coming out is the other double helix. So it starts with 1 double helix, you end up with 2. That's happening in every cell in your body right now. It's a bit mesmerizing, that as well. I'm just going to let that run. Now, The second thing that DNA does, which makes it more important than any other molecule, is that it contains information. It is a code. That after Crick and Watson used that data to elucidate the fact that it was a double helix, it took many people, dozens of people, led by Francis Crick, but the first people to crack the notion of the code were these two, Heinrich Mattei and Marshall Nirenberg. They did it earlier than 68. This photo was taken in 68. And what they began to work out was that those letters, the arrangement of those letters on the inside of the double helix was a coded alphabet. So each one of those A, T, C's, and G's were arranged into threes called codons, and each one of those threes encoded an amino acid. And amino acids are strung together in the cell to make proteins. And all life that has ever existed is made of or by proteins. Right? So DNA is the code. It doesn't actually do anything else apart from replicate and contain the code, but it codes for proteins, and proteins do everything that life requires. So it was Francis Crick that came up with what is known as the central dogma of biology, which applies universally to all living things, as far as we know, that have ever existed, that have been alive, which is that DNA makes protein via an intermediary molecule called RNA. It's called the central, another ridiculous historical aside, but it's called the central dogma, and dogma is a word that we don't really like in science. Not since the Enlightenment. We've tended to avoid the word dogma and leave that to organized religion. And Crick was once asked in the 70s why he chose to call it the central dogma. I'm not aware of whether this is a joke or a genuine statement because I've only ever read it. His response was, I didn't know what dogma meant. Right, so just careful how you name these things. Anyway, so the point of that history, this is undergraduate biology I've just described, the history of how life works. The point of all of this, the point of working out how genetics works, how DNA works, ultimately continues to be, to try and understand not just how inheritance works, but to attempt to deal with the fact that there are diseases, inherited diseases. And this is, I'm skipping forward, decade by decade, giving you the slightest overview of how this works. When I talk about the code, we have about 3 billion letters of code in every one of our cells. We have arranged into chromosomes, 23 pairs of chromosomes. This is what they look like during a particular part of the cell cycle. And so, again, skipping over billions of dollars and millions of man-hours worth of research, instead of looking at individual traits like pea color or individual diseases or individual characteristics that humans have, the great drive from the 1980s until the beginning of the 21st century was to try and understand how human genetics causes disease. And so rather than looking at those individual characteristics, we decided to look at our entire genome, to read our entire genome in one go, the Human Genome Project, which I think is probably the greatest scientific endeavor that mankind has ever attempted until the frigging Large Hadron Collider. It was the most expensive until the Large Hadron Collider, we just understand humans or understand the universe, whatever. So, after billions of dollars and thousands of man-hours and dozens of labs, largely funded by the Wellcome Trust. In 2001, the first draft of the human genome was published in this journal, Nature, and also in another journal, which I forget the name of. Now, it was the first draft. It was a complete readout of the 3 billion letters of genetic code that each of us carries. I was working in a lab at the time. I was working on a gene on chromosome 14 involved in eye development, and I remember the press announcement earlier in the year. Bill Clinton had stood on stage and said things like, we have read the language of God, which is a bold thing to say regardless of your religious beliefs. And I remember the news report from the BBC at the time saying that within months we would have understood and maybe even cured all human genetic diseases. And then they cut to a kid with Duchenne muscular dystrophy looking very hopeful. We have cured no diseases since the Human Genome Project was completed in 2001. That is not a criticism of it. It's a criticism of the press. This is the process of science. This is how we discover things. What happened in 2001 and in the subsequent years as we began to understand how human genetics, or we began to explore human genetics, is we found out two things. We found out how little we know about human genetics. That was the main thing, right? So we like to think ourselves as complex organisms, which we certainly are. And when we think about that central dogma, we think about traits in pea plants being passed from generation to generation. There was an assumption that because we are complex, that you would find a lot of genes, that we have a great number of genes. And what the first thing that we found out, which was very surprising, was that we have fewer genes than rice. This is a water flea called Daphnia. This is a roundworm, and this is a banana, which was a great surprise, right? We have about 21 or 22,000 genes, and that doesn't go nearly far enough to explain how complex we are as organisms based on our old model of genetics. The second thing we found out, which is a big surprise, which was that of the 23 pairs of chromosomes that we have, the 3 billion letters of code that we carry in us, almost none of it is genes at all, right? Less than 2% of our DNA is actually genes. And so the question that emerged soon after that was, well, what the fuck is the rest of it? And again, the press characterized this as a great crisis in genetics, that this was something that, we'd just been wrong for the longest time. When actually it's good science, right? We sometimes describe this as the Rumsfeldian type problem. We didn't know what we didn't know. And the last 10 years of human genetics, I think, have been finding out what we didn't know so that we can then find out what we need to know afterwards. He's a great philosopher, Rumsfeld. So so that's the breakup of the human genome. So this tiny wedge here is genes. These two great majorities are bits of DNA that aren't genes. This very slim wedge here is bits that were once genes but have evolved into being not genes anymore. So basically what it said was that we actually don't have a really robust understanding of how human genetics works at all. We don't really understand how inheritance in humans works. For diseases such as Huntington's disease or Duchenne muscular dystrophy or cystic fibrosis, which are inherited in a very linear, direct way, like pea plant traits, got a total understanding of that. For anything more complex than that, it's really, really difficult. And that's fine. That's a good scientific problem. It means that we spend the next 10, 15, 20, 100 years working these things out, largely funded by the Wellcome Trust. So, we learn this stuff there's a sort of cultural problem here, because we learn this stuff. We learn about how characteristics are inherited, and we learn about this at school. So, who can tongue roll? Right? I learned that at school, that's an inherited trait, that I can't remember whether it's recessive or dominant, but if your parents can tongue roll, you can tongue roll. It's not, right? You can learn how to tongue roll. Lots of genes are involved in it. So whoever can't tongue roll and is mocked by your family, you can tell them that it's not genetics at all. Eye color. Everyone knows that brown eyes are dominant over blue eyes. So if you've got a brown-eyed parent and a blue-eyed parent, you're going to have you can predict the proportions of how many blue eyes and brown-eyed children you're going to have. No, you can't do that at all. There's a green-eye gene, and what that means is there is a full spectrum of colors from very pale blue to almost black-brown, and it is almost impossible to predict what color eyes your children will have based on the eye colors of their parents. There are some characteristics which you can predict. This is a form of polydactyly, 6 fingers. I come from Suffolk. And this one, that's earwax. There are two types of earwax. Sticky and dry. And it's encoded by a single gene, and you can predict with high degree of accuracy what type of earwax your children will have based on what type of earwax you have as parents, which kind of puts a different spin on designer babies. Anyway, so this is all good. This is good stuff. This is good science. It's a good way of finding out how genetics works. It's a good way of trying to understand the immense complexity of human beings. That to think that things as sophisticated as us would be answered by models as simple as what works very directly in pea plants was silly, right? So working out what the rest of the genome does, also a major problem and a major project in genetics that's been running for the last 5 years. One of many projects that has been going on for ages now in the last 10 years for genetics, working out what the rest of the genome does. Results published. This was this project called ENCODE. The results were published earlier last year, at the end of last year, and established that huge chunks of the non-coding, the non-gene-based DNA that we carry around in us was doing something. We don't know what that something is, but it is active in some ways in many, many cells. And again, not a scientific crisis, but a good scientific problem to get hold of. What is it doing? We don't know. Come back in 10 years' time and I'll still be standing here saying the same thing. So I'm a journalist, and this is something. The complexities and the sophistication of human biology is something that is quite difficult to convey in short-form journalism. This is a nice quote from H.L. Mencken, the sage of Baltimore, the editor of the Baltimore Sun for many years during the first half of the 20th century. For every complicated problem, there is a solution that is simple, direct, understandable, and wrong. So one of my favorite games is Googling scientists discover the gene for X. And you come up with millions of headlines and web pages from every publication across the world where scientists have discovered the gene for a specific thing. I sort of want this to be Rutherford's rule, but just in a really vain way, that the answer to that, if the headline says, scientists discover the gene for X, they haven't because it doesn't exist. So here's some of my favorite examples. I can whip through these so we can get to the next more interesting bit. I write for The Guardian. Scientists discover the gene for cocaine addiction. No, they didn't, because it doesn't exist. What they did discover is a higher frequency of a particular variant amongst a broad population that associates with a particular behavioral type, right? And that's the same for pretty much all of the things I'm about to show you. It isn't a gene for anything, because there are almost no genes for things. There is a gene for sticky earwax. There is a gene for cystic fibrosis. Almost everything else, there are many, many multifactorial ways of getting to that particular behavior. Behavior is really, really complex, right? Oh. Now, admittedly, soft targets going after the Daily Mail, but this is quite fun. Transsexual gene, no, it doesn't exist, and they didn't discover it, and no scientist said it. The gene that can scare you out of your mind, the gene that makes you left-leaning, the love cheat gene, 1 in 4 born to be unfaithful, claim scientists. The scientists didn't claim that at all. What's super good fun about those last 3 is it's all the same gene as well. So, I mean, imagine having that combination. Just absolutely devastating. Here's a good example, right, of why this can't possibly be true. And now, I have to be straight with you. The nuance in the article is much more acceptable than the way the title is, the way the headline is written. Scientists discover height gene. Right, now, I think this is a good example, because height is an absolute measure. Right? There's one number for height. I'm 5'10", right? And 5'9½". But so that's one measure. But of course, it's made up by dozens of things that you could easily imagine. It takes no thought to see how the fact that there's one number involves lots of different factors, like the lengths of different bones. I could stand next to another 5'10" man, and we would have entirely different proportions. I could stand next to someone who was very genetically related to me, but had been raised in an entirely different environment, an entirely different diet, and done different exercise during their life, and they wouldn't be the same height as me. So one measure, right, doesn't mean that it's that one gene is responsible. Oh, that was a few years back, but it goes on, and it never goes away. Is there a gay gene? Well, of course there isn't a gay gene. This was fun because is there a gay gene was answered by two people, neither of whom were geneticists. They are gay people, but who aren't possibly the best people to address questions of genetics. Here's another one, a study that will, a gene that predicts what time of day you'll die. That was in November last year. Not much use if you get hit by a bus. Right, that's all fun, right? So there is a serious point to this massive misreporting and misunderstanding of the sophistication of human genetics. Telegraph reported a gene that makes you good at taking risky decisions. The gene involved here is called monoamine oxidase, and it was associated in this study with high-risk behavior, I think in city folk. A New Scientist reported it as the warrior gene because it also has a different variant, has a high frequency in Maoris. Now, this is a guy called Bradley Waldrop, who photographed in 2009 wearing very distinctive orange clothes. A couple of years earlier, he had returned home after a night of heavy drinking, and he shot his wife's best friend, I think six or seven times, and killed her. And with a machete, he hacked his wife into a few pieces, although amazingly didn't kill her. Now, he was sentenced to death. And in 2009, at this trial, his lawyer successfully argued that he had the same variant of this gene, monoamine oxidase, as Maoris have. He has the warrior gene. And therefore, he was predisposed to this kind of behavior, and therefore, his sentence could be moved from death to life. Now, I'm not a supporter of the death penalty, but that decision was made on really bogus scientific grounds, a real misunderstanding of the complexities of genetics. This is a quote from one of the jurors, so one of the people making that decision. Diagnosis is a diagnosis, a bad gene is a bad gene. This is a deeply, deeply problematic societal issue that we are facing at the moment. And that was 2009. This happened in the last few weeks, right? So Adam Lanza and his spree killing in Newton. Well, the state examiner after that incident was ordered that his genome be sequenced in order that we might find something interesting and something significant about why he perpetrated this act of evil. And what we will find out from looking at Adam Lanza's genome is nothing at all, unless it turns out he has cystic fibrosis or polydactyly, or something which is very clearly identifiable in his genome. We've been doing this for years, by the way. This is not new. It's just new technology. On the left, you've got John Wayne Gacy, and on the right, Peter Curtin. John Wayne Gacy killed 33 teenage boys in the 1960s. Peter Curtin was known as the vampire of Dusseldorf. He killed 9 people in the 1930s, although it's thought that he killed 60, probably 60. After both of those guys died, their executed, their brains were taken out for examination to determine what it was about the structures of their brains that turned them into serial killers. Behavior is complex and will not be explained by genetics. Elements of genetics will explain various physical and psychological behaviors, but not in any way that could be diagnostic. What it is phrenology. This is the idea that the shape of your head, a popular Victorian idea, the shape of your head would determine your behavior. Right, I'm going to change gear again because that's a bit morbid and I'm running out of time and I haven't even got to the interesting bit. So we go back to this, right? So we're thinking about DNA, and we're thinking about how DNA, this universal language via the the central dogma, Francis Crick's misnamed principle. All living things since for the last 4 billion years, all uncountable species, all even more uncountable numbers of cells have carried this code, this information, and passed it from generation to generation. As I said earlier, when looking at the Habsburgs, the way we understand genetics in general is by looking at families, but also by looking at farming, right? Which is the opposite of natural selection. It's artificial selection. We are choosing traits in individuals and breeding them with other individuals in order that the next generation has those characteristics and has them in an enhanced way. Now, once we worked out that the genetic code was universal, and once we invented and discovered the technology which allowed us to do this, because that code is universal, because it's shared by all living species, in the 1970s and 1980s, we began doing something which farming previously couldn't do, which was to skip out the breeding step. What I'm talking about is genetic modification, right? We could take the genes or the DNA from one species, and we could put them into the genome of another species with whom previously we were limited by farming or by breeding. Okay, there are millions of examples of this. We've been doing this since the 1980s with world-changing repercussions, not just in farming, but significantly the Human Genome Project was largely based on this type of technology. My favorite example is this. So this is Freckles. Freckles is a goat. Freckles lives on a farm in Utah, and though almost entirely goat, Freckles contains the gene that encodes spider silk from the orb weaver spider. And it's been engineered in such a specific way that when you milk, when Freckles lactates, she produces spider silk. So if you milk her, you take the milk out, I've done this, the only goat I've ever milked happened to be a spider goat. Right? You take the milk out, and then the fibers, which are proteins of spider silk, which are short, they're short fibers, but when you raise them out and they're exposed to oxygen, they form long links, which is the silk that these spiders use to swing from when they're falling. It's terribly vain to show a photo of oneself, but I do this for a point. I wrote an article about Freckles for The Observer last year, and they felt the need to caption this like this. Really? Okay, so why would you do such a ridiculous thing? Well, spider silk is one of the most interesting materials that we know of. It has higher tensile strength than steel. It has it's an extremely desirable thing for us to use, right? And we can't farm spiders, right? Because spiders are cannibalistic, right? So you put two spiders next to each other, you end up with one spider. So what you do is you take something that you can farm and you introduce the gene for that. It's an extraordinary form of breeding. The last time that these guys had a common ancestor was 500 million years ago. And until this point, until genetic engineering, farming was about mating things. And it's probably not wise to think about the sexual union of a spider and a goat because it's not possible. But we can now bypass that. We can breach the species barrier in order to create products that are useful, that are considered useful for us. Here's another example, which I like. This is a slightly out of focus photo. This is a type of cress. And this cress has been genetically engineered so that if it grows in the presence of landmines, it turns purple, right? So these ones, there's a mine at the front here, there's no mine at the back here. So the idea being that you take a handful of the seeds, you scatter them in a field where you think there are mines, where there's a purple flower, where there's a purple plant, there is a landmine, where there's green, there is not. For reasons that are not quite clear to me, this has never been commercialized and has never been used, but that's the reality of science in the real world. This is a jellyfish, Aquara. It glows. This is not a genetically engineered jellyfish. This is what the jellyfish does in the sea. It glows green. at the beginning of the century, scientists identified the protein that actually causes that fluorescence, that with breathtaking lack of imagination that scientists sometimes display. Can we get that, maybe? Hello? Shall I? It's all right. It's fine. It's green, it's a protein, and it fluoresces, so they called it green fluorescent protein. So once that was characterized, a Nobel Prize for that discovery in 2009, that became an incredibly important tool in the toolbox of genetic engineering, because it means that you can attach this as a tag to any sort of modification that you want to do, and you can see it. Sometimes genes are very cryptic. The way they're expressed, the way they behave in cells or in organisms is something you can't always see very easily. But if you put a sodding great fluorescent tag on it, you can see it, exactly what it's doing, as soon as that animal is born. So there have been millions of examples of this, because it's used all the time. This one is one of my favorites. This is from 2011. And that cat has GFP, green fluorescent protein, expressing in almost all of its cells. And again, not just because it's awesomely cool. This creature was created by AIDS researchers who used that tag to follow a protein that fights HIV as it's distributed across the body. So incredibly striking, incredibly beautiful, but also a really important tool for understanding HIV and AIDS. Now, that's genetic modification, sometimes called genetic engineering. What has happened in the last few years is that this field has moved on. It's gone through a step change. I was doing experiments not dissimilar to this 10 years ago, but in order to do that, it took thousands of man-hours and a PhD and years of experience in order to do something as artisan as that. And what has changed is there's been a new movement in genetics, but also in other fields that surround genetics, that recognize that DNA, this universal tool, this incredibly powerful set of tools that evolution has provided, are things that are incredibly useful for us, not just for farming, but for doing genetics, for doing human genetics, for working out how diseases work, for producing drugs, for producing fuels, for addressing environmental change, and so on. The problem that was identified by a new breed of scientists who call themselves synthetic biologists was that you have to be artisan, you have to be a skilled practitioner of genetics in order to do this. And what synthetic biology is about, distinct from genetic modification, is about doing, right? It's about creating, it's about doing, and specifically it's about engineering. So it's kind of a shame that genetic engineering has the word engineering in it, and synthetic biology doesn't, because engineering isn't really a science. Apologies to any engineers in the audience. Engineering is about applying science in order to address specific questions. And that's what synthetic biology is. So what happens at the beginning of synthetic biology, this is in 2000, is you start writing out, scientists start beginning to think about genes, not as DNA, but as components, right? As components in circuits that follow logical patterns. And this was the first real synthetic biological circuit, which looks, if any of you are familiar with electrical engineering, it looks a lot like an electrical engineering circuit. But in fact, what these four elements here are genes. This one being GFP, the one I've been talking about. And what this relatively simple circuit does, I can demonstrate using you as an audience. And this doesn't always, I've done this a couple of times before, and it really doesn't always work. But I need 3 volunteers who are capable of standing up and down on demand. So I need someone from, thank you. No, you need to be at your chair. And maybe someone, thank you very much, and maybe someone at the back. Not on a stool, because that's dangerous. One more person. There you go, brilliant. Okay, now how this works, this is called a negative feedback loop, right? So when this gene is on, right, its action is to turn this gene off. And when this gene is off, it means that this gene is on, right? And when this gene is on, follow the path round, it means that this gene is off. So when this gene is off, on, off, on, and it's because there are 3, it's continuous. So if you could stand up, and wait, who's the next person? And you stay sitting, and you could stand up. Now, when she is standing, you have to sit. And when he is sitting, you have to stand. And when you are standing, you have to sit. And when she is sitting, you have to stand. Go. Go. So you got to stand now. You got to stand now. There you go. Go. That's right. Okay, brilliant. Keep going, keep going. That's a negative feedback loop, right? So it's continuing. No, I said keep. Keep going. I mean it. This doesn't stop for you. Anyway, right, this negative, no, it's broken. All right. This negative feedback loop is inserted into bacteria, right? And so there's no output there. You loop round and you'll keep doing that forever, or until you get bored, right? But what you need is an output in order to measure what's happening. So in the circuit, it's GFP, right? The same green fluorescent protein. So when this one's on, this one's off. When this one's off, this one's on. When this one's on, it switches that one off, but it also activates GFP, right? So every time this one's on, the cell glows green. Because that takes, there's a time delay between those things, which is the same time delay as when these 3 people were standing up and down. GFP comes on and then it goes off, and then it comes on and it goes off. And what you have is a cell which glows, but it pulses, right? So it goes boom, 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 boom. It does it in about a 15-minute cycle, because that's how long these genes take to cycle through it. Now, if you really want it, we can actually do that, but we need a 4th person who is willing to sing. Right? Come on, come on, come on, for fun, come on. You look like you might want to do it. Right, so your job, thank you, kind volunteer. Anyone, I was pointing at you. anyone who wants to sing. Right, thank you. So all you need is one note, right? Just sing one note, and you only sing that note when he is standing, okay? So every time it loops, he's going to stand and he's going to sit. You only sing when he's standing. Everyone else closes their eyes. I mean it. This is exactly how genetic synthetic biology works. Go.
If I've got my eyes closed.
No, you don't close your eyes. Right. Thank you. Hopeless. I mean, really, that was terrible. But that's the principle. Right. The problem. There's so many problems with that. It's unreal. The problem with this back in 2000 is that in order to engineer a circuit like this, you still need artisan levels of skill in genetics, in genetic engineering, and years of experience. We sometimes refer to this as the plug socket problem, right? So, when anyone travels, you've got to take with you a bunch of adapters because different countries have different plug sockets. And that's what it was like, and that's what it continues to be like in straightforward non-synthetic biology genetic engineering, which is that it's difficult to engineer 2 genes from 2 different species together so that they can talk to each other in such a way that they produce the protein that you want. So in about 2004, the BioBricks Foundation founded and identified this as a fundamental problem, right? And so what they did is, their principle is to standardize DNA components, genes, to make them universally interchangeable in exactly the same way that electrical engineering did many, many years ago. So that if you want to build a circuit on a circuit board, you don't have to reinvent the diode every single time you do it. You buy a diode from Maplin's and you stick it on a circuit board and attach it to a thyristor or whatever, because the parts plug together. And that's what BioBricks intended to do. So that circuit that I showed you a minute ago with you guys failing to stand up and down and sing at the same time, this is, I think, the high point in synthetic biology, and this is from 2011, so almost 2 years ago. This is what the circuits look like now, which looks like an electrical engineering circuit. All of those parts are interchangeable, but they're arranged in such a way. I'm not showing this in order to understand it. I'm showing you this because to show the complexity of where our circuitry is in synthetic biology right now. And what this does, rather than having an oscillator, which means that you sing or GFP comes out every 20 minutes, this identifies and destroys cancer cells, right? So it has a logic to it, which is Boolean, so it effectively works as a computer. And what it does is it's a virus. All this circuit is packaged into a virus, which you infect into. This is in cell culture, so it's not actually in organisms yet. But you introduce the virus to cells, some of which are cancerous, and the virus infects the cells, and then it basically asks that cell 5 sequential questions. And if the answer to all 5 of those questions is positive, then it is identified in electronic engineering terms as a true value as a cancer cell, and at which point it activates, which is the output here, it activates that cell's suicide program, right? So it infects a cell, asks it 5 questions. If the 5 answers are positive, then the cell kills itself. If any of the answers to those 5 molecular questions are negative, then the virus leaves and the cell is fine. So this is only in cell culture, right? It's being tested in animals right now. But if you think about how we treat cancer now, which is still the main 2 therapies are chemotherapy and radiotherapy. These are blunderbuss approaches, right? We kill as many healthy cells as we do cancerous cells because we have very poor ways of identifying the difference and getting rid of one rather than the other. This is much more like a sniper, right? Because it acts on a single cellular level. So if you introduce this to a group of cells, it will only kill the cells that are dangerous. Very early days. All of the components in this circuit came from the BioBricks Foundation, a catalog, who have built a catalog of parts. There are currently 10,000-ish parts in the BioBricks registry. And they're characterized and they're detailed and they're freely available to people who registered and wish to understand this process. And this is exemplified by an annual competition that they've been running for the last few years, which is called iGEM, the International Genetically Engineered Machine Competition. And the competition is for undergraduate students who, during their summers of their second years, they construct a problem and they use parts from the registry to construct a solution to that problem. They have about 8 weeks to do it. And this has been happening since about 2005. And I think about what I did during my PhD only 10 years ago, which took me 3 years to get one gene into one cell. And these guys are taking multiple genes from multiple organisms and putting them together in ways not just experimentally, but in order to create things which have specific engineering applied processes, right? So the winners this year, it sounds trivial, but it's pretty cool. They designed a circuit and put it into bacteria that if you you can seal it in between 2 pieces of plastic or paper, and if you put it inside a meat tray in a supermarket, they will change color when the meat begins to go off, right? So that's very practical. The team from University College London had a much more ambitious and equally implausible, but they made a lot of progress towards this, which was that many of you will be aware of the problem of microplastics floating in the ocean. There are tons of tiny particles of plastic floating throughout the oceans of the world, which enter the food chain and are extremely damaging. They designed a genetic circuit that went into a bacteria that would process those plastics and aggregate them into lumps, their ultimate goal, unachieved, but it's a good aim, was to build an island based on the aggregated plastics floating in our ocean. And this is the sort of ambition. This is the sort of idea that is behind synthetic biology as a field. And I think there's a lot of hype that surrounds this field, which I'm sort of party to now, but I think there's a lot of enthusiasm, and this is going to be of great significance in the forthcoming years. The skill level is a really important thing. These guys are undergraduates, right? They're not PhDs in genetics. They're not professors of anything. Many of them are not biologists. I went to the UK meetup back in September before the final competition, which happened in November. There were 85 students there, and 30 of them were not biologists. They were not studying biology in any way. They were engineers, mathematicians, and a couple of humanities graduates. God forbid. It's a joke. But it means that the skill level for entry is greatly reduced. This, of course, is the pathway of all technology. I sit here pressing a button in order to forward the slides with no understanding of even how the button works, let alone the code behind it or how the projector works. But someone invented that a long time ago, but I don't need to know that. In a similar way, the ethos, which is something you can argue about behind this, is that they don't need to know how DNA works. They don't even need to know the history. They don't need to know about Misha and Rosalind Franklin and Nirenberg and Mattei. What they need to know is whether it works or not, because they can design something and then they can build it. Fitting the ethos, the grand prize is this, a giant aluminium Lego brick. All right, I've been going on for bloody ages now. Can I keep going? All right, I've only got 4 more slides. Right. Now, you can't talk about genetic engineering without coming across this guy. This is Craig Venter, 2010. He published this. This is Mycoplasma mycoides JCVI-SYN 1.0, much more easily remembered as Cynthia. The first synthetic organism, kind of. Right? There's a lot of complexities to what he produced and how it was reported in the press, something that I care about, not just as someone who was a geneticist, but also someone who is a journalist. Was it a synthetic organism? Kind of. What it was that he'd taken the genome of a live cell, Mycoplasma mycoides, which by coincidence is a goat pathogen, something that infects the udders of goats and causes minor form of mastitis. And what he did is he read that entire genome, fed it into a computer, and then got a DNA sequencing machine to produce its entire genome, took that genome and put it inside the chassis of an existing cell. So what it was the first cell since the origin of life that wasn't born of another cell, right? Whose parents were not an existing cell. And that's an enormously significant thing to do. That's not what I have this up here now for. He did something interesting in there, which is the final part of this sort of ramble about what DNA does and what it can do. All the way through this, I've been referring to DNA as a code, right? And that's what it is. It's a data storage device. It's a data storage device which has an enormous and deep history. 4 billion years, we've been passing on data down every single cell, all the way to every single cell in your body. You can trace the history of life on Earth through any single cell that you have on you right now. So you start thinking about this as a data storage device rather than as a biological entity. It's very stable. It's got a 4 billion year history. Not only that, but we're very good at reading DNA from cells that have been dead for a long time. We've sequenced, just this week, we sequenced genes from Richard III in the Leicester car park to the extent that we could demonstrate that he was Richard III. Was it Richard III? Yes, right, thanks. And that's only 500 years ago? A few hundred years ago. We've also sequenced the entire Neanderthal genome from 40,000 years ago. And that's we've done mammoths from 60,000 years ago. And I believe the record holder is somewhere between 450 and 800,000 years ago, and it's a saxifrage, which is a type of perennial plant that some of you will have in your gardens, but has been buried in Greenland ice for all of that time. So it's very stable, right? You know, it's a really stable form of data storage. It's also very dense because it's molecular and it's very small. Venter, as part of that, this cell, in order to demonstrate, partially in order to demonstrate that this cell had not been born from another cell, he also, partially because he's kind of showing off, he encoded some secret messages, some encrypted code in there for people to discover. Easter eggs is what we call them if you're a gamer. So there were several Easter eggs hidden in the genome of this little cell here. One of them was the addresses of the names and addresses of all the people who involved in the project, 42 of them. One of them was a URL. You had to solve the code. You had to crack the code in order to read the rest of it. And the third one was 3 appropriate stroke pretentious quotations, depending on which side of that stroke you fall on. And one was by Oppenheimer, from the Manhattan Project. One was from James Joyce. And one was from everyone's favorite physicist, Richard Feynman. And it was this is the sort of mantra of synthetic biology. What I cannot build, I cannot understand, except the real quote is this. What I cannot create, I do not understand. There's no reason to show you that other than just a bit of prodding of hubris. You know, accuracy in DNA sequence is really important because it's the difference between having cystic fibrosis and not having cystic fibrosis. And so, anyway, he got that fractionally wrong. And the Joyce quote as well was he was immediately issued with a cease and desist copyright infringement letter from Joyce's estate. But anyway, that's by the by. Many of you will have seen in the last, this was in 2010, many of you will have seen in the last week that there was a big news story from the EBI, the European Bioinformatics Institute in Cambridge. This is Nick Goldman. And what he's holding there is a tube of dried DNA powder that contains encoded into it all of Shakespeare's sonnets, an HD video of Martin Luther King's I Have a Dream speech, the PDF of Crick and Watson's 1953 paper describing the structure of DNA, and a whole bunch of other information. I can't remember how much information is actually encoded in this. It's several gigabytes. Do you remember? It's gigabytes. Now, the density of this, because it's molecular at its level, is such that the estimates has been a cup of DNA like that could store 100 million hours of HD footage, right? Are you nodding because this is Wellcome funded as well? Probably is again. Now, the code itself, what's really interesting about this is that we have now got to the stage where our understanding of DNA and our ability to manipulate it means that we can absolutely eschew the code that all of us, that all living things have ever relied on, that central dogma. We can write our own code. Venter did it. The code was not particularly sophisticated. This is a much more sophisticated version of that code. Why would we do this? Well, because it's dense. It's also permanent, right? It outlasts magnetic tape, which is currently how some places like CERN store their data. It's much more dense than Blu-ray or DVD. There's a better reason, which is that this is a format that will never, ever not be studied, right? There is never going to be a time in human history when we're not going to be interested in DNA. And therefore, there's never going to be a time that we're not going to be able to have the technology to read it. You pass a quarter-inch magnetic tape to someone now, and almost no one has the technology to decode that, right? You take a Blu-ray. I've got a Blu-ray disc which doesn't play laser discs. Soon, DVDs will be redundant. People are going to be looking at DNA forever. Now, this is again, proof of principle. It's very early stages. It's useless for writing memory now. It takes a long time to write the memory. This particular sample here was made in Cambridge to test it. It was sent to Germany in an envelope and then decoded. And of the many gigabytes, which I'm sorry to say I've forgotten the actual number, when they decoded it, it had an error rate of 0. It was 100% accurate of all the data that was stored in there. And well, that's it really. I mean, this is the story of life on Earth. It has been going on for 4 billion years, over the last 100 years. We've worked out a lot about how it works, but we've got a long way to go in terms of understanding how the complexities of life and humans and how they relate to diseases. But we've also got to the stage where we have such sophistication and such knowledge, without meaning to be too hubristic, that we can actually use the tools of evolution to address the biggest problems that humanity currently faces. And that's all. Thank you very much. And you can buy that from April the 4th. Please do.
So you're going to come up and have a chat?
Oh, yeah, sure.
Yeah.
Questions? Yes, thanks. Right, questions.
Have a seat.
So the reason there's 2 books there is because halfway through writing, I realized that these are 2 parallel stories. That our understanding of how DNA works takes us to a point where, took us to a point where we could ask 2 fundamental questions, which is one, how did it start, where did it come from, but also, what can we do with it? And as I was writing, I realized that they're very related stories. It's the same technology, it's the same industry involved, but actually they're profoundly different things. And so my editor at Viking decided that we should publish 2 books. And this is that's the front cover, and this one is the front cover. So they're inverted back on each other, so you can read both in either direction, and it'll confuse some people, and that's it. Questions?
That's it.
Do you have any questions? Shall I? I can sit. Yep.
Well, thank you so much for that. I've just got a couple of kind of observations or remarks to make before we open it up to questions. So hold your thoughts. We've got about 15 minutes. So
went on for ages.
plenty of time. No. Also, because we've got too long for questions, if anybody does want to give us any feedback or ask any questions after this, then the wireless network here is Lighthouse Arts, and the password is submarine. You're Adam Rutherford at Twitter, and we are Lighthouse Arts. So if there's anything that we don't get to talk about, then please go online and talk to us like that. I guess what really struck me in the second part of the presentation, Adam, is some really interesting alignments between what's happening at the kind of outer reaches of the synthetic biology community and what a lot of people in this room will be quite familiar with, which is hacker culture and maker culture, which is, kind of introduced this very kind of DIY, kind of can-do approach to, building, sort of code, kind of making electronics. And there's a very, very kind of interesting kind of, I suppose, resonance between what's happening in, I suppose, technology space and what is starting to happen within biology space. Is it something that you kind of can see as well? Do you know of there being,
It totally already exists. So biohacking is a thing, right? So it's to do with entry levels to the technology. The reason that hacking exists, well, the origin of hacking is coders who are really interested in the intricacies of code. But the reason we have hack days and stuff is because the technology is transferable and you don't need to be quite so expert to do it. And that's where synthetic biology is going. So I talk about iGEM as a university undergraduate projects thing, but actually they've just launched iGEM schools, right? So, if any of your teachers, I would thoroughly encourage you to get your children, your pupils, children, involved in this space. I've seen 13-year-olds doing genetic engineering that would have taken me 5 years, 10 years ago, and that might sound terrifying. I'm aware that is something that you might look at and just go, holy crap, why do we allow this to happen? I think it's something that I don't think it's dangerous. I think with the types of experiments, the types of demonstrations that they're doing, it doesn't have potential danger. I'm very aware of the fact that is there is the dual technology problem behind that, which is that things that are neutral can be made to have maleficent properties. I think it's something that we can't avoid. I think it's something we should encourage, and I think it's something that we should encourage with. We should watch it carefully. But in the same way that there are probably hackers in this room, certainly within a mile of here, who could bring down Western civilization if they really wanted to. That doesn't mean we stop coding.
Just checking. Few interested looks. I was really struck.
They're not going to admit it.
But, you know, they're showing their remarkable restraint, you know. Well, I was also really struck by your, you know, kind of your comment that, you know, kind of the UK, the current UK iGEM team, you know, kind of around 35 of them are non-biologists.
Yeah.
We've got mathematicians, engineers, and, you know, kind of humanities students. And what's interesting, I suppose, for us, you know, kind of who come from a sort of a kind of an artistic bent or a media art bent, is that artists have been quite kind of active in this space for a long time. We were talking earlier today about the Tissue Culture and Art project, who are effectively kind of growing, you know, using biology as a sculptable material. We had a project here a couple of years ago called Lab Life, where we had artists doing astrobiology experiments on fruit flies with geneticists. So, you know, this feels like a very kind of current and sort of quite urgent kind of conversation within art. And what's interesting in the art space is that they are very, very happy to confront those ethical issues. In fact, a lot of the work is designed specifically to kind of test the kind of ethical parameters, you know, involved in working with life. And are you starting to see a similar kind of, you know, kind of almost, I suppose, kind of critical design kind of thread running through early synthetic biology experiments?
It's an incredibly difficult, it's a minefield to navigate. And it's a minefield for a number of reasons. I'm aware, I'm very aware of the fact that we're talking about things which seem so fundamental, and especially when I stand up there and talk about the toolbox of evolution, which has been around for 4 billion years. You know, that's a sort of powerful idea I'm trying to use. And then I'm talking about, also in the same sentence, using it to describe things which are either important or a little bit glib, right? You know, they're things that hackers do on their spare time, weekends, school kids. So I'm very aware of that sort of weird dichotomy. But I think the reason that I'm doing that, and I think the reason that I think that's an interesting space to be part of, is to kind of to kind of disabuse people of the idea that it is fundamentally wrong, right? And rather encourage the notion that we should be thinking about how powerful they are for beneficence, and look at the potential for maleficence, so that we can have informed conversations about where those lines can be drawn, right? And the GM debate in this country was lost before it began, and was skewed by, oh God, I'm going to regret saying this, but was incredibly skewed by people with political agendas which didn't necessarily relate to what was actually possible and what was actually available at the time. And as a result, we have a slightly peculiar situation, which is in Europe we have no genetically modified foods in our diets, whereas in America and in the rest, in fact, I think in the world in total, the estimate has been that there have been, I think it's 80 trillion genetically modified meals consumed in the last 10 years. Right. Now, I'm not making any sort of value judgment or any political or agendaed statement about those two facts. What I am saying is that these conversations have to happen in an enlightened society in a agenda-free and educated way. And I think you only do that by having as many of the facts on the table in front of you in an educated and informed public as possible. And I don't know how you do that other than talking about these things openly.
That feels like a very good moment to open it out. But can we have
That was cruel.
Can we have some lights on so we can see you? I know I saw one hand up. Tom, did you still have a question? Maybe more lights on. So, Yep.
I was really interested when you brought up your pie chart of like, if we've got a lot of like redundant genes going on, or stuff that's kind of maybe useless or
Yep.
not doing anything,
Yep.
can we not use that to store other people? I mean, how much
Well,
space might we have?
Well, it's yeah.
My own USB drive.
I've been doing this for a while, and I've never come across that question before. Total hat off there. It's a fun question to ask. The question's wrong, and the reason is because when you say it's redundant, there was a time when people thought they referred to it as junk DNA. That was the stuff that didn't encode genes was once referred to, was once labelled as junk DNA. That's a very powerful title because it's something that people can really get hold of. It didn't, in the scientific community, amongst geneticists and molecular biologists, it lasted about 30 seconds because we knew right back in the early 70s that it wasn't junk at all. It wasn't that it was doing nothing, it's just that we didn't know what it was doing. And there's a big difference between those two things. Is it doing anything? You don't know. What's it doing? We don't know. So it is one of the great points of that study on ENCODE was not to work out what it was doing, but to show that it was doing something. The complexities of human genetics are such that I would be extremely anxious about introducing any data storage into a human, because the repercussions would be extremely unpredictable.
Compressed what was there. It's probably really badly compressed.
It's your coder. I've reached the limits of my understanding of code. No, good for thinking about it, though. Because in fact, so when I talk about at the very end, when I was talking about the storage of the sonnets and the Martin Luther King, there was another version of this was done in September by a group in America, led by a guy called George Church, and they used a very straightforward binary, which was where there's an A and a T, that's a naught, and there's a C and a G, that's a 1. And the version I was talking about was sophisticated to the extent that I really don't understand it at all. But they had an error rate. They had an error rate of 5 parts by 5 errors per petabit, which is pretty low. But the new version with the UK version had a 0 error bit amongst the same amount of data. So it's levels of code. I think that when the reporting of this was done, the press release that came from EBI said that there are 3 zettabytes of data in the world. I think a zettabyte is 10 to the 21, so which is 10 with 21 zeros after it. DNA as a storage mechanism is really only useful at using this technology as an archive because you can't access it very easily and you can't write it very easily. But you can start somewhere.
Another question at the front here?
Yeah. When this was talked about, well, at least what I heard about it, was that what had previously been considered to be the junk was actually epigenetic switches. And so at that time, it seemed like the path that was going to pursue that was the one that was going to get to dealing with health issues. Whereas what you just showed us at the end about the virus that goes into cells and checks out whether they're cancerous.
Yep.
That looks like a much faster route.
Yep.
So,
well, it's a fair point. You've identified a thing. It's very interesting that you spot that, right? Because basically, the cancer thing is the high watermark in synthetic biology. It's also the only high watermark. Right? I talk about it because it's amazing. It's amazingly powerful. There's a lot. It's something which we need to do a great deal more work on identifying more of these sorts of circuits. The reason it's so successful as a biological computer is because the cancerous cell that it identifies is the most studied cell in the history of biology. It's called the HeLa cell, which causes a particular type of cervical cancer. It's got a great history to it, which is fascinating if any of you know it. It was derived from the cervix of a dirt poor black woman in Baltimore in the 1940s who died subsequently of the same disease. But as a result, that cell was studied and has existed in labs ever since. We know more about cancer and genetics as a result of that cell than maybe any single other thing. So, I showed you something which was very precise and successful because we know an extreme amount of information about the thing that it was targeting. To get to that stage that I showed, in any other cancer, and in an actual organism, rather than just in cells and culture, is probably decades away. So thank you for asking that, because I now have my caveats to explain. It's good that we do that, right? Because I'm giving you the top line, I'm giving you the headline. I'm being a journalist about it. What was the first part of it was, understanding the epigenetics. Well, yes, that's what ENCODE is all about. Epigenetics is the study of stuff which is above genes, it's beyond genes, and those are the modifiers. Those are the things that are not involved in protein production that modify, that control the way that we actually behave as organisms in terms of cellular function. So the very fact that we have too few genes to explain how complex we are as biological organisms, what the rest of the genome is doing, as explored by projects like this, not just this, but like this, is how we begin to address why there are too few genes. Because there clearly aren't too few genes, right? Because we work. There are too few genes because our model was wrong. Right behind you.
It's not a question, it's a statement. You mentioned the woman in America, that woman.
Henrietta Lacks.
Yeah. Anybody who wants to read about her, I recommend. I think it's probably called The Invisible Life.
It's called The Immortal Life of Henrietta Lacks.
It's a fantastic book. I just like
by Rebecca
everyone to know
Skloot.
that they should, yes, absolutely. They should read it. It's
It was the
fantastic.
best science book of last year. It's a breathtaking story.
I'm
Yeah.
not a scientist, but I loved it. I thought it was absolutely.
Heed her words. It's a monumental story.
Alex.
I'm an artist, but my original background is electronic engineering.
Nice.
So
Were you getting like really warm feelings when I was showing those?
I felt comfortable. But when you basically understand like Boolean algebra and all that kind of stuff.
That's good because I don't.
I was just wondering, how do you actually ask a cell? It's a question. How do you ask a cell a binary question, a yes or no question?
Well, there's two answers to that. The first answer is the binary question is something that I probably can just about handle. But basically, if a molecule is on, which is exclusive to a cancer, and you can detect that, and it's exclusive in the sense that if it's not on, the cell is not cancerous, then you do have a binary state. The second answer is the Z et al. Science 2001. I can send you that paper tomorrow if you want. I mean, it's incredibly complex, and I mean, look at the circuitry there. I don't know. I don't understand it, and I'm sort of mates with the lead author on that. I'm not sure he understands. Sorry.
One more question.
Yeah, when we see these stupid stories about, gay genes and all that kind of nonsense, how much is the scientific community implicated in that stuff coming out? Because all scientists, like all of us, need financial support and need to find financial input. I'm interested to know who's...
Are you a scientist?
No, not at all. Well, I studied science, but I'm not a professional scientist. How much of the problem is the PR agency of the Science Institute, just getting it wrong and saying any publicity is good, and how much of it is to do with the desperate need for funding? What's causing those stories to come out?
Well, it's a good, valid question. Hello? Oh, does that? And it's an incredibly complex ecosystem of an answer. And what you've just described is part of that problem, that we scientists need to generate publicity in order that the public are interested, because almost all money that doesn't come from the Wellcome Trust.
Try not to mention it.
Is that it? Have I used up my credit there? Right, sorry. Comes from the public purse, and therefore you need to engage with the public in order that they understand the questions that are being asked. So that is one of those factors. I don't think it's the biggest factor. There is another factor, which is that the whole system of scientific discourse doesn't reflect the way that science is done. Right? There is a huge cultural problem in terms of how science is disseminated to the public, which has a sort of catastrophic gap between what scientists are doing on a daily basis. So if you think about it, right, we get grants for a period of time, for 3 years, to fund a PhD student and a postdoc to answer a specific question. And during that time, you come up with a major result that you publish in an academic journal. And the academic journal publishes that, and because it's a major result, it comes with a press release, which comes from the journal and might come from the university. And the press pick that up, and they publish that in a newspaper or a magazine, and makes it onto the news, and then subsequently might make it onto a documentary. Right? And every stage of that process is a potential diluter. But at the bottom of the process is the fact that science doesn't work like that, knowledge doesn't work like that. Right? It doesn't work in 3-year gaps, it doesn't work in grant periods, it doesn't work in staff periods. That's not how we acquire knowledge. The publication process is extremely biased against the way that we acquire knowledge. We have to draw a line in the sand at one point and say, this is now ready to release to the public, as if it's finished, as if we have an answer to this problem. Now, I describe that in a sort of utopian way. It exists because it has to exist like that, because you can't spend all of your time telling people that we've found this out and now we're going to find this out. And imagine doing a pitch for the Large Hadron Collider 50 years ago, where you say, well, I need about $200 billion to simulate the origin of the universe. And them sitting there with their checkbooks going, and when are you going to find that? You're going to go, Genome. Right? Same with the Human Genome Project. And I say this I forget the fact that the Wellcome Trust is sitting in the front row here. One of the great benefits of having organizations like that and scientists willing to engage in multidisciplinary international collaborations that don't have specific answers to them, where the outcome of the Human Genome Project is we have found out a shitload of stuff that we didn't know. Right? That's really, really important. So the whole ecosystem is flawed because it has to be. Mary wants to make a point.
No, I want to ask a question. Would you rather that there was interest and excitement around science in the press, even in society, even though the reporting of it isn't exactly accurate, rather than the opposite, which would be rather than people don't hear about interest?
No, absolutely not. What I would we need excitement. We need these grand gestures as well. ENCODE was criticised. It was profoundly criticised, much by geneticists when those publications came out, because for various reasons, but a lot to do with the PR about it. And I felt that a lot of that criticism was misplaced, because I believe that we need grand gestures. I think we need things like the Large Hadron Collider, and we need the Human Genome Project to get people excited. And even if they're esoteric and small studies, like the DNA storage device that was all over the press last week, it's a tiny preliminary study. Whether we ever use that again, I don't know. I think we probably will, which is why I like to talk about it. But it gets people talking about it, and that's really good. It's not a rather, it's not an either or. What I would like is that we engender a society which is scientifically literate enough to be extremely excited by these types of things without the need for headlines that misconstrue, misrepresent, have shock value with no scientific backing to them. PR, psychon, this is what I do, it's my living. I don't want to spend my time telling people that the Daily Mail is a rubbish newspaper. What I want to spend my time doing is telling people why science is fucking awesome and why the whole of civilization is built on science. That's what I want to do. The reason I put this up, in answer to your question, was this is an example of a GWAS, a genome-wide association study, which is one of the things that came after the Human Genome Project, which was when we began to look at not just the first human genome sequence was based on 6 individuals. So it was a very broad example of a generic human. And of course, everyone knows that we're 99.99% similar in our DNA, but we are all absolutely unique. And in terms of the number of letters of genetic code that we have, that number is, we are unique through our past and future, unless we live for 10 trillion years. What happened after the Human Genome Project was we began to look not just at individual genes and broad differences between people, we began to look at small differences between lots of individuals. So what this is the result of taking a population of people, a huge number of people, that all have the same, in this case, disease, and looking across their entire genome to see what similarities and differences they have. Rather than looking at individual genes which we think are involved in that disease, you look at all of their genome. So it's like a scattergun approach. What is going to stand out, right? And in this example, which is a type of muscular dystrophy, what you see is that there is one particular bit that stands up here, right? This happens to be a gene in the HLA region, which is to do with our immune systems. This is the type of study, an immensely complex study, as you can see from the fact that it just looks complex, that the press might report as being this is a gene, and this is a gene that relates to a specific disorder that all of the people, every column in this graph, have, right? So this is a gene which is closely associated with a particular disease. Brilliant. This red line here is a significance level. So below that, it's not considered significant. Above that, it is. Right? This one is for schizophrenia. Right? So I don't have the pictures, but I could produce 30 headlines from the newspapers which say the gene for schizophrenia in the same way they did for other diseases. Right? I show this again not so that we can understand it, but just to show what a mess human genetics is, a good mess, a scientific mess, because here's a significance level which is higher than it was for the previous one. And what do you find? These are all people with various degrees of a complex disorder like schizophrenia. And what you find is you have 30 odd individual places, some of which relate to genes and some of which don't, which are all variable across people who have what we consider to be one syndrome, actually a range of a spectrum of disorders. And what that means is it's complex. It really requires a lot of deep statistical analysis to try and understand the etiology of this, how that disease actually manifests. What it doesn't show is that this one here, I don't know what MAD1L1 does, right? But it doesn't show that this is a gene for schizophrenia or any of those things. It doesn't show that any of those genes are genes for schizophrenia, which is how it gets reported. What it shows is that there are a proportion of people who have schizophrenia who have an increased likelihood of having this particular variant in our human genome. So, genetics is extremely complicated.
On that note, I'm sorry we're going to have to bring this to a close, but we've been treated to an incredibly insightful kind of journey through the human genome. An amazing way to start our Brighton Science Festival program that continues with David's residency. We're going to do a hack day with young people, probably no biobricks yet, but we're going to work on that as we go forward. And we've got lots more, so check out our website for more information about that. But please help me thank Adam very much for what was an amazing talk.